Developing PIGE into a rapid field screening test for PFAS. The development of a spectroscopic total fluorine technology to screen for PFAS would revolutionize the environmental investigation and remediation field, just as the advent of handheld photoionization detectors for volatile organic compounds has now become standard practice. We have developed a laboratory method for total fluorine measurements called particle-induced gamma ray emission spectroscopy, or PIGE for short. And this limited scope CERTIP project was to design how this laboratory method could be made into a useful field deployable tool. There are many potential uses of such a screening method for PFAS, but I will focus on the obvious one here. If you're going to look for a lot of water samples to determine which are contaminated and which aren't, there's a significant time and cost involved to collect and analyze those hundreds of samples via traditional methods. A rapid screening test, which could pinpoint which samples are non-zero at the wellhead in the field, could save significant time at site characterization or remediation monitoring. Simply put, if there is no fluorine, then there are no PFAS in that sample. Instead of having 2 to 5 site visits to the various wells to sample groundwater over 1 to 2 year period, we could accomplish this all in one visit. This alone would save millions of dollars per DoD facility to quantify, assess, and enable remediation planning. This type of approach for rapid screening could reduce labor and analytical costs during remediation efforts in a similar manner. Since PIGE has already been shown to measure total fluorine on the surface of any solid substrate with good sensitivity, this project assessed the feasibility to adapt this technique into a screening technique for total fluorine, especially for aqueous samples in the field. First, we determined the best beam energy to use for the protons, which defines the minimum size of the accelerator to use. Then we optimized the target preparation methods, developing a new solid phase extraction method to increase our sensitivity to fluorine in aqueous solutions. By going with flow-through filters and a higher sample volume of 2 liters, we were able to get sensitivity for any standard PFAS analyte below 50 parts per trillion within a 3-minute measurement. Since both of these steps were successful, we invested in a new accelerator design that would allow this laboratory-based technique to become field deployable. Specifically, we altered the design of an existing accelerator from Starfire Industries in Champaign-Urbana, Illinois, which originally designed and built a prototype field deployable accelerator with DARPA funding for Homeland Security purposes. And we have proposed modifications that will make it ideal as a field deployable system for PFAS screening. It will produce a high yield of 4 MeV protons, which when coupled with our detector system and new felt filters, will be able to return total fluorine results in the field within minutes per sample. Now we have the opportunity to directly measure fluorine in groundwater samples with a van that can move around sites and DoD facilities. We propose to build a prototype system to increase our technology and manufacturing readiness in anticipation of a fleet of such vehicles.